Today's topic for discussion is genetics, monohybrid cross. Genetics deals with inheritance. It deals with inheritance. And what is inheritance? An elephant giving birth to a baby elephant. When we plant a mango seed, obtaining a mango plant are examples for inheritance. The similarities between brothers and sisters also represent inheritance, whereas these brothers and sisters of the same parents show dissimilarities also. This is an example for variation. Both are considered in genetics. Both aspects are considered in genetics. Our ancestors also know about both inheritance and also variation, but they don't know the scientific basis for the phenomena. The scientific basis for inheritance and variation was for the first time revealed by Gregor Mendel, an Austrian monk teaching biology. He selected Pisum sativum garden pea plant for his experiment. It's a common name. Pisum sativum is the scientific name. Garden pea plant is the common name. The plant has certain advantages. It is most suitable for experiments in genetics. The advantages associated with this plant are this. The pea plant is annual. This is annual plant. It has perfect characters, well-defined characters are there. It has bisexual flower. Each flower produces both male gametes and also female gametes. Bisexual flower is there, and it is normally self-pollinated and can be easily manipulated for cross-pollination. So, this is normal self-pollinated plant species of family Fabaceae, and after sexual reproduction it produces large number of seeds. These are the advantages associated with selecting the particular plant. The pea plant is annual plant, and many generations can be studied in short span. It has perfect and well-defined characters. It produces perfect flowers or bisexual flowers capable of producing both male and also female gametes. This is normally self-pollinated, can be easily manipulated for cross-pollination, and it produced a large number of seeds. So, all types of permutations and combinations due to recombination of genes can be studied from a single cross-pollination. Now, the methods employed by Mendel, were partly contributed to his success. What are the methods? He applied and also mathematical logic for study of biology for the first time. He used statistical data, mathematical logic, and also applied mathematical principles, math principles for the first time. This contributed to his success to a great extent, and each time he focused on a single trait. Now to the characters chosen by Mendel for his experiment. Mendel has observed many characters in Pisum plant. He has chosen 14 plants differing in 7 pairs of characters. With regard to height, he found some plants are tall, whereas others are short, dwarf. With regard to color of the flower color, violet, and other flowers are white. With regard to color of the fruit pod, green, whereas its contrasting trait is yellow. With regard to color of the seed, some seeds are yellow, whereas other seeds are green in color. With regard to position of the flower, some flowers are axial in the axils of leaf, whereas others are terminal in location. With regard to shape of the fruit, inflated, the other contrasting character is constricted. With regard to shape of the seed, some seeds are round, whereas other seeds are wrinkled. He has chosen 7 pairs of contrasting characters. A tall plant and a dwarf plant are dissimilar only in their height. They are similar in all other characters. A violet flower producing plant and white flower producing plant differ only with regard to their color, and they are similar in remaining six other characters. Whereas the green pod producing plant and yellow pod producing plant differ only in this trait, and they are similar in remaining characters. So Mendel has chosen these characters, 7 pairs of characters. He studied the inheritance of all the 7 pairs of characters independently, and let us begin with the monohybrid cross or inheritance of one gene, the pattern of inheritance of single gene in pea plant. Single gene is called monohybrid cross. The study of inheritance of a single gene is called monohybrid cross. Mendel used a pair of contrasting characters with regard to height. He has selected tall and dwarf plants as parents. Tall plant and dwarf plant were selected as parents. The tall and dwarf plants were true breeding plants. True breeding plants. True breeding means they produced, tall plant produced only tall plant, whereas dwarf plant produced only dwarf plant. This is due to repeated selfing. Repeated self-pollination is the reason for true breeding nature of any plant. In this case, the tall plant always produces tall progeny, the dwarf plant always produces dwarf progeny, and one of the plant is chosen as female parent, the other is chosen as male parent. In the female parent, he has removed stamens. The removal of stamens is called emasculation. He collected pollen grains from the male parent and transferred them to the stigma of female parent. So hybrid is prepared. The seeds are collected, and when seeds are planted, first filial generation, first F1 generation is produced. All plants were tall only. All plants were tall only. He has chosen the parents, tall, true breeding tall, and true breeding dwarf. When they were crossed, the seeds are collected, and when they were planted, all hybrids are tall only, and he wanted to investigate what happened to dwarf character. So he self-pollinated. He self-pollinated the F1 hybrids. He self-pollinated the F1 hybrids. The reason for this is he wanted to know what happened to dwarf trait of the parent. When after self-pollination he collected the seeds, and when seeds were planted, F2 generation was raised. F stands for filial generation. So, the F2 generation was raised. To his surprise, and the tall plants were there, so also dwarf plants in the ratio of 3:1. The dwarf character did not appear in hybrid, but it reappeared in the F2 progeny, that is second filial generation. And basing on this experiment, he formulated through two important principles in genetics. One is the law of dominance, and the second is the law of purity of gametes. And what were his observations? Let us consider what were the observations of Mendel. The first, the traits are factors, genes. The term used by Mendel for genes is factors. Factors appear in pairs. Factors appear in pairs. How he came to that particular conclusion? The tall parent produced the tall factor in its gamete. The dwarf parent produced dwarf factor in its gamete. Both are there in the tall hybrid, F1 hybrid, but dwarfness did not appear on selfing. Because the dwarf factor is there in the hybrid, it reappeared in the progeny in F2 generation. So, it is clear that the factors appear in pairs. And so, to express that, we can use TT, capital T for dominant character. The character that appears in hybrid is a dominant character. The character that did not appear in hybrid condition is recessive character. This is represented by small case letter TT. Because both factors are there in the hybrid, it must be capital T and small t. So, F1 plants are hybrids because they contain the factors for both tallness and also dwarfness. Whereas on selfing, 50% of the gametes produced by the hybrid contained capital T factor or a tallness factor, whereas remaining 50% gametes contained the factor for dwarfness, and because of their random combination, the F2 progeny is tall and dwarf both will be there in F2 progeny, and the proportion of tall is 3:1 to that of the dwarf. And this can be because it is producing two types of gametes. By using a Punnett square, the different phenotypes and genotypes can be observed. So, on selfing it will produce two types of gametes. Half of the gametes will carry capital T, remaining half will be carrying small t, and these are the male gametes. The female gametes also will be similar. Half of them will be carrying capital T, remaining half will be carrying small t. So, these are the gametes, 50% capital T, 50% small t, 50% capital T, 50% small t. So, on the random fusion, you have capital T, capital T, capital T, small t, capital T, small t, and small t and small t. Basing on F1, whenever both are there, whenever capital T and small t both are there, the plant will appear tall only. So, it has both capital T's and automatically it must be tall. Regarding the other one, because there is one capital T, this plant also must be tall, and here also we have same combination, it also has capital T and small t, this must be tall again. And since there is no capital T here, only two small t's, this must be dwarf. So, this square is called Punnett square. This device is helpful in predicting what are the phenotypes and possible genotypes in a cross. So, 50% of the gametes are having capital T, remaining 50 small t. When they fused randomly, we have this kind of result. When he carried the same kind of monohybrid cross with other traits of the pea plant like violet flower crossed with white flower, all F1 were violet. In F2, he obtained violetish to white in 3:1 ratio. Similarly, with axial flowers and terminal flowers, when he carried out this, the F1 progeny were all axial flowers. In the F2 progeny, they appeared in 3:1 ratio. This is true with other characters also. When he conducted experiments on shape of the seed, round seeded plants crossed with wrinkled seeded, all F1 were round. In F2, he obtained roundish to wrinkled in 3:1 ratio. Mendel obtained 3:1 ratio not only in the monohybrid cross of tall and dwarf plant, but with all remaining 6 pairs of characters also, he obtained similar result, indicating that something universal and fundamental is involved here. And the result, F2 result ratio in case of axial and terminal flower is 3:1. In case of violet and white flowered plants crossed, also the F2 result is 3:1. Round seeded plant crossed with wrinkled seeded plant also, the F2 result is 3:1. Similarly, the yellow seeded crossed with green seeded also, the F2 result is again 3:1, and the green pod crossed with a yellow pod also, yellow podded plant also, the result F2 result is 3:1. So, it directed him to formulate his first principle called law of dominance. Law of dominance. What is law of dominance? First, the factors or traits appear in pairs. Factors appear in pairs. It is capital T in homozygous condition, small t, or capital T and small t, and these combinations represent what is called factors appearing in pairs. When different factors of the same character, say tall and dwarf together, only one of them appear, and that character is called dominant character. That character is called dominant trait or dominant character. So, when both tall and dwarf are together in the F1, only tallness is exhibited but not dwarf character. So, that is exhibited, that expressed itself is dominant trait, that was suppressed is called recessive trait. And in this case, tallness is dominant trait, whereas the dwarfness is recessive trait in this case. Dwarfness is recessive in this particular case. With regard to other characters, yellow color of seed is dominant. Seed is dominant, whereas green is recessive. With regard to location of the flower, position of the flower, axial flower is dominant and terminal flower is recessive. With regard to flower color, violet color is dominant, whereas white color is recessive. With regard to green is dominant over yellow color of the pod. With regard to seed color, we have considered seed color here, and with regard to, we have seen flower color, seed color, pod color. With regard to position, we have seen axial versus terminal. With regard to the other character, that is shape of the pod, inflated condition is dominant over constricted condition. Constricted condition is recessive, whereas inflated condition is dominant. With regard to shape of the seed, round shape is dominant over wrinkled shape of the seed. Round shape is dominant over wrinkled shape of the seed. So, with all 7 contrasting traits, Mendel came to similar results or similar conclusions, indicating that the law of dominance is working in each and every case. Let us go back to the original monohybrid cross between tall and dwarf plants again. Regarding the monohybrid cross between tall and dwarf plants, we have P generation, parent generation, tall crossed with dwarf plant. Both are true breeding plants. They produce, tall produced only tall progeny, dwarf produced only dwarf progeny. When these are crossed, seeds are obtained and the seeds all are tall. All F1 progeny, all seeds of the hybrid developed into tall plants only. And this here it is cross, whereas here it is selfing is done. The plants, F1 plants are selfed and the seeds are collected and planted. The F2 generation is obtained, and the F2 progeny we have tall and also dwarf in the ratio 3:1. The tall and dwarf factors are together in the F1. Let us write the factors in capital, dominant factor in capital letters and recessive factor in small letters. In hybrid, both capital T and small t are together. But in F2, there is no intermixing or blending of characters. We have the plants either tall, completely tall, or completely short. There is no plant in between. Let us take one more example. Let us take the color of the yellow color of the seed. This is dominant, whereas green color of the seed is recessive. If we consider that seed color yellow crossed with green, and in F1 all plants produced yellow seed only. On selfing in F2 again we have yellow is to green in the ratio 3:1, and there is no mixing, there is no color where you can see half of is yellow and remaining half is green, or any other kind of blending is there. The case is true for violet also. In F1 only violet, and in F2 we have both violet and white, not any kind of intermixing. So, this suggests this points to another fundamental aspect, that law of purity of gametes. The second principle is law of purity of gametes. The first principle is law of dominance is the first principle, whereas law of purity of gametes is the second principle. This suggests that in heterozygous condition, in F1 hybrid, both factors, both genes capital T and small t are together in the hybrid. During gamete formation, either capital T enters the gamete or small t without any kind of blending. If we observe Punnett square, the picture will be more clear and lucid. The F2 plants are tall and their physical trait, their appearance is tall, and inside it is capital T and small t. This is hybrid. This is subjected to selfing. The F1 is subjected to selfing. The F1 is subjected to selfing, and we obtained F2 result, where because it is hybrid, it produced 50% of the gametes having capital T, remaining 50% having small t, and there is no intermixing, and that is what is called law of purity of gametes, also called law of segregation. So, during gamete formation, capital T, the gametes contain either capital T or small t, but never both. So, the progeny here is tall, and again this is capital T and small t, the progeny is tall again. Here it is capital T and small t, the progeny is tall again. Here it is small t and small t, the progeny is dwarf. So, during the formation of gametes, though the F1 hybrid has both capital T and small t, the gametes contain either capital T or small t, never both. This is what is called law of purity of gametes, otherwise called law of segregation. This is the second principle Mendel derived basing on his monohybrid cross, otherwise called the inheritance of single gene. So, two important principles, two important conclusions were derived by Mendel basing on his inheritance of one gene experiment. Law of dominance. Using the law of dominance, Mendel was able to predict that F1 hybrids show only one trait, that is dominant trait. Using the principle law of dominance, Mendel could predict that only dominant trait will be seen in the progeny of F1. And also, using the same principle, law of dominance, Mendel could predict the F2 progeny will be in the ratio 3:1, 3:1. Dominant will be 3 ratios and recessive will be 1 ratio. And here, what is this tall and dwarf? Both are called alleles. Both are called alleles. These are slight variations of the same gene. These are slight variations of the same gene. Both influence or both affect the character called tallness or height. Similarly, violet color of the flower and also white color both affect the color of the flower. Similarly, seed color, all these are alleles. These are slight variants of the same gene. And in sexually reproducing organisms, the alleles will be present, and both alleles, if they are same, like true breeding tall, it will be TT, capital T, capital T, whereas F1 will be capital T and small t. True breeding dwarf will be 𝗬𝗏𝗧. And if both alleles are same, then it is called homozygous. The condition is called homozygous. It is the case of capital T and also the case of small t and small t. Both alleles are same, then this is called homozygous condition. If both alleles are different, both alleles are dissimilar, different, then they are called heterozygous. It will be capital T and small t represent heterozygous condition, and capital T or small t condition refers to homozygous condition. All gametes have, this will produce all gametes with capital T inside, this will produce all gametes with a small t inside. Whereas in this particular case, heterozygous condition, 50% of the gametes have capital T, remaining 50% of the gametes have small t. So, heterozygous individuals produce different kinds of gametes, 50% of one kind and remaining 50 of the other kind. Now, if Mendel applied binomial theorem to predict the result with regard to other monohybrid cross experiments, and the F2, the F1 progeny, the F1 hybrids are heterozygous, that is TT. Half of the gametes, this is subjected to selfing, this is subjected to selfing. 50% of the gametes contained capital T, remaining 50% of the gametes contained small t. This is true for both male gametes and also female gametes. And to obtain the result, he has squared the possible gamete types, and he obtained 1/4 capital T plus 1/4 capital T small t plus 1/4 capital T small t plus 1/4 small t. Since these two are same, we can write it like this: TT 1/4 capital T and 2/4, that can be reduced to 1/2 capital T small t and 1/4 small t. So, basing on this application of binomial theorem, we can draw certain conclusions. What is the probability of getting the capital T 0.25, or otherwise this we write as 1/4 TT. What is the probability of obtaining small t? That is also 0.25, that is 1/4 small t. What is the probability of obtaining heterozygous condition? That is 0.5, that is 1/2 capital T small t. So, from looking at the Punnett square for F2 progeny, so 50% male gametes have capital T, 50% female gametes also have capital T, remaining 50% female gametes have small t, remaining 50% male gametes have small t. So, this is TT and small t. This is dwarf, this is tall, this is also tall, this is also tall. What is the phenotypic ratio here? Phenotypic means what is visible. The phenotypic ratio in this case is 3:1, or still better, we can write as 3/4:1/4. Out of 4 results, 3 will be tall and one is dwarf. So tall is to dwarf in the ratio 1:4 or 3/4 is to 1/4. This is what the progeny will appear, what they look like. But what is inside? What is inside? To find out what is inside, the Punnett square comes to help, and let us write the genotypic ratio. Let us write the genotypic ratio, that is capital T, capital T. How many of the progeny are like that? 1 or 1/4. And how many progeny are small t are heterozygous? This is half the progeny are like that. 2 or 1/2. How many progeny are homozygous dwarf? 1 or 1/4. So this can be written like this. The genotypic ratio for monohybrid cross, where complete dominance is seen, is 1:2:1. This is homozygous dominant. For homozygous dominant, homozygous recessive, homozygous recessive. Regarding heterozygous progeny, it will be half of the progeny will be like this. So capital T will be one of the 4 ratios, whereas capital T small t will be 2 of the 4 ratios, whereas small t will be one of the 4 ratios. So these are the conclusions drawn by Mendel. He continued his experiment. He planted the seeds. He selfed the T2 dwarf plants of F2 progeny. He selfed the T2 dwarf plants that contain small t factors, and he obtained F3 also dwarf and F4. On selfing F3, he obtained F4 also dwarf. Next, when he planted tall plants, when he selfed these tall plants, he obtained again in F3, he obtained 3:1 tall is to dwarf. He obtained 3:1 tall is to dwarf. When F2 tall selfed, the F3 progeny were 3:1 again tall is to dwarf. So he wanted to know which tall plant is homozygous and which tall plant is heterozygous. To know that, he devised another test. He devised another test called back cross. He devised another test called back cross. F1 hybrid is crossed with parent or parent type. The F1 hybrid is crossed with parent or parent type. The F1 hybrid is tall. This is the phenotype. The genotype is capital T small t. This is crossed first with the dominant parent type. Since it is heterozygous, it produces 2 kinds of gametes. It produced 2 kinds of gametes. And since this is homozygous, it produced only 1 kind of gamete. This produced only T, whereas this produced 50% gametes capital T, remaining 50% gametes small t. And here the progeny is all are tall. All are tall, but homozygous tall to heterozygous tall is 1:1. Or we can write this result in this manner also. For a backcross with a dominant parent, backcross with a dominant parent, backcross with dominant parent, we have 𝗬 dwarf 2:0. The 𝗬 progeny will be 2:0. Mendel observed that in the F2 progeny, dwarf plants, in the F2 progeny, dwarf plants always produce dwarf plants. So when the F2 progeny, dwarf progeny is selfed, F3 were also dwarf, F4 were also dwarf. So for any number of generations, the progeny was dwarf only. Whereas this kind of tall plant produced only tall, whereas this progeny produced, this F3 progeny produced tall:2 in the ratio 3:1. This tall, when selfed, again produced tall:2 in 3:1 ratio in F3 and subsequent generations also. To identify which is homozygous tall, which is heterozygous tall, Mendel devised what is called backcross, and backcross is crossing with either parent or parent type. To know genotype of the organism, this cross is normally employed. So the F1 hybrid TT, the F1 hybrid capital T small t was crossed with a dominant parent type capital T capital T. Since this is heterozygous and it is homozygous, it produced two types of gametes, it produced one type of gamete. So this is the gamete produced by dominant parent type. These are two kinds of gametes produced by the F1, capital T, capital T, capital T, small t, all are tall. So 2:0, tall:2 dwarf progeny is in the ratio 2:0. When such cross is made with a recessive parent, F1 crossed with recessive parent type, when F1 is crossed with recessive parent type, this is the possible result. Tall, this is F1 tall, so this is capital T small t crossed with small t dwarf parent. Since this is heterozygous, it produced two kinds of gametes, and this is homozygous, this produced only one kind of gamete. So this is the Punnett square for that. Capital T, 50% gametes contain capital T, remaining 50% gametes contained small t, and all gametes contained small t only. This is capital T, small t, small t, small t, and this is its genotype, and its phenotype is tall, its phenotype is dwarf. Its phenotype is dwarf. So what is the ratio of tall:2 here? Tall:2 here is 1:1. Tall:2. In case of this particular backcross, we have equal number, that is 1 half of the progeny will be tall, remaining half will be dwarf. Whereas in the other type of backcross with the dominant type of parent, all progeny are tall, there is no dwarf progeny. What is this particular kind of cross with a recessive parent? This is normally called test cross. This is also a back cross. A test cross will reveal the genotype of the individual. A test cross will reveal genotype of the individual. So, to identify whether the plant is homozygous tall or heterozygous tall, to identify whether the plant is homozygous tall or heterozygous tall, back cross with recessive parent is carried out. This particular cross is also called test cross. By testing, it will tell what are the genes inside. That is the reason for calling this test cross. So, to identify whether an individual is homozygous for a particular character, homozygous or heterozygous, this cross is employed. The application of the other type of cross is in agriculture, where to make the hybrid homozygous for dominant trait, it is repeatedly crossed with dominant parent type. Its application is normally in agriculture, whereas its application is to judge whether the individual is homozygous tall or heterozygous tall. So, this is the application of the application and use of backcross. Let us look at the board to use the knowledge obtained in this discussion. This is EAPCET question basing on our present content. According to Mendelism, which character is showing dominance? Is it terminal flower, green colored seed, or wrinkled seed, or green pod color? Here, this is the correct option. The answer must be 4. This is recessive, this is also recessive, this is also recessive. So, fourth option is the correct one. If we look at the EAPCET 2002 question paper, this is the question. A gene is said to be dominant if. When a gene is dominant when it expresses itself in both homozygous and also heterozygous conditions. So, this is not the correct option. It should express itself both in homozygous as well as in heterozygous conditions. So, this is incorrect, and this is also incorrect, and a combination of the two. It should express itself in homozygous and heterozygous conditions. So, this is the correct option. Gene normally has its expression. So, the correct answer is 3 in this case. If we look at NEET 2014 question, what are alleles? Alleles are different molecular forms of a gene. The first option is the correct one. So, answer in this case is the first one. Regarding a NEET 16 question, a true breeding tall P is crossed with a true breeding dwarf P plant. When F1 plants are selfed, the resultant genotypes are. So, about the monohybrid cross, the question is based on genotypic ratio. The genotypic ratio is 1:2:1, and both options have 1:2:1, but we should go for homozygous tall, heterozygous tall, and dwarf. So, homozygous tall, heterozygous tall, and dwarf, this is the correct one. Though it has the same ratio, it is representing, the first one is represented by heterozygous tall, this is incorrect. So, we cannot go for the second option. So, the first is the correct and most appropriate option.